Bismillah ar-Rahman ar-Rahim. Just to recap with the atherogenic lipoproteins, we mentioned that they include LDL. This is the most common type, but they also include the IDL and the VLDL and the LPA, the special LDL-like particle which we talked about. In addition, we have the chylomicron remnants. So these are the atherogenic lipoprotein, but the most important is the LDL. These atherogenic lipoproteins accumulate in the bloodstream like that. So let us say the LDL as representative of the atherogenic lipoproteins. Then many factors contribute to changing the properties of the endothelial lining of blood vessels, so that this contributes to the accumulation of lipoproteins particles in the intima. This is the tunica intima of the blood vessel, and this is influenced by different factors, including some hemodynamic factors like hypertension, for example. Hypertension is a risk factor for atherosclerosis, and it hemodynamically contributes to the trauma to the endothelium, so it causes injury to the endothelium. Also, the increased number of lipoproteins in hyperlipidemia in itself may affect the properties of the endothelial lining. Other factors include some toxins in smoking. Anyhow, the LDL now accumulate in the subendothelial intima and they become minimally oxidized. So lipid oxidation takes place. This is because of the accumulation of some reactive oxygen species from the oxidation in the vascular tissue. This can be exaggerated by some oxidative stress induced by such factors as smoking, for example. So smoking can be a contributing factor to the oxidation of lipoproteins. So the lipoproteins which are oxidized that way, we call them minimally oxidized lipoprotein or minimally modified lipoprotein or LDL, which represent the lipoproteins here. So smoking contributes to the oxidative stress, but smoking has many other effects, and this is why it is a major risk factor for atherosclerosis, as we are going to mention later. So once the LDL becomes minimally modified by the oxidative stresses, it's going to stimulate the endothelium of blood vessels. So minimally modified LDL exhibits a potent biologic activity capable of inducing the endothelial cells to express adhesion molecules. So the endothelial cells start to express adhesion molecules for monocytes, for example, XLAM. And also they express monocyte chemotactic protein, MCP1, for example, and also macrophage colony stimulating factor. So these are the factors which are expressed by the endothelium after they are being induced by the minimally modified LDL, modified by oxidation, of course. So key word for the pathogenesis is the oxidation of LDL and the other atherogenic particles. The expression of these and other molecules results in the recruitment of blood monocytes. So monocytes start to get an access into the subendothelial intima as well. So they enter and they are acted upon by the monocyte chemotactic factor. So they are more and more recruited into the intima and they become differentiated into macrophages. So this is the sequence of events. First, we have an injury to the endothelium by hyperdynamic factors like blood pressure, or by toxins, for example, from smoking, or by the increased number of LDL in hyperlipidemia. So they start to gain an access into the intima, and they become minimally oxidized by the active oxygen species produced by the tissues, and this is exaggerated and contributed to by smoking, for example, which induces more oxidative stress. So the minimally modified LDL will stimulate the endothelium of blood vessel to express adhesion molecules, for example, XLAM, and to express monocyte chemotactic factor, which recruits the monocytes towards the intima, and then they become differentiated into macrophages by the macrophage colony stimulating factor, which is produced by the endothelium as well. Then further oxidation of the minimally modified LDL takes place by reactive oxygen species derived from macrophages together with lipooxygenases of the macrophages. So this augments the oxidation of LDL, and now we have more oxidized LDL. We call them OX-LDL. Could be any other atherogenic particle as well. At the same time, the macrophages express scavenger receptor, which is going to bind to these oxidized LDL species, and they are internalized inside the macrophages, and the macrophage eventually becomes stuffed with these oxidized LDL species. Unlike the LDL receptor, the scavenger receptor is not downregulated by high level of cellular cholesterol. If you remember, when the LDL is internalized into liver cells or in other nucleated cells, the cholesterol coming from their degradation is going to determine the level of intracellular cholesterol. It can inhibit the de novo synthesis of cholesterol, and it can inhibit the expression of the LDL receptors. This doesn't happen here. The scavenger receptors do not possess this regulatory property. So the macrophages become laden with oxidized lipoproteins without any limit, without any kind of regulation. So they will end up in the subendothelial spaces stuffed with oxidized LDL and other atherogenic species of lipoproteins. So now the macrophages are transformed in what is called foam cells, and they appear under the endothelium like a fatty streak, and this is the early lesion of atherosclerosis, which we call fatty streak. These macrophages secrete inflammatory molecules and cytokines. These stimulate the proliferation of smooth muscle cells. So the smooth muscle cells undergo proliferation, and they are recruited to the site of the lesion, and some of them may be converted into foam cells by ingesting oxidized LDL as well. So foam cells represent the hallmark of fatty streaks, and they contribute to the development of advanced lesions by the production of cytokines and growth factors like macrophage colony stimulating factor and interleukin-1. Then fatty streaks may advance to more advanced lesions, which we variably call atheroma. Sometimes we call it atherosclerotic plaque, which could be stable or unstable, and it is variably called fibrous cap. The macrophages produce proteases, and these proteases are going to cause fissuring of atheroma, and this will lead to exposure of the blood to subintimal collagen and tissue factor. And this is going to contribute to the development of thrombosis. We have to notice that smoking has many roles in the development and advancement of atheroma. Apart from its pro-oxidant role, it contributes to the reduction of high-density lipoprotein, and also it contributes to the depletion of vitamin E from the lipoprotein particles. Usually it is present in the shell of the LDL and other lipoproteins. Also, smoking contributes to thrombosis by activating platelets, and the activated platelets are going to produce platelet-derived growth factor. And this growth factor has a role in the proliferation of the smooth muscles. So we can say that the effect of smoking as a risk factor is due to its pro-oxidant effect, and also because of depletion of high-density lipoproteins, and because of depletion of vitamin E from the shell of the lipoproteins, and also because it activates thrombosis and activates platelets, so that platelets produce platelet-derived growth factor, which contributes to the proliferation of smooth muscle cells.